Today we will discuss about platelets, their formation, functions and variations. So platelets, these are the non-nucleated blood cells. They are spherical or oval in shape. Size, it is around 2 to 4 microns. These are colorless cells and the count, it ranges from 1.5 to 4 lakhs per mm cube. Now you can see this is the ultrastructure of platelets. So what can you see there? You can see some glycogen particles, microtubules, microfilaments, some granules are there. So electron microscopically, Golgi apparatus, mitochondria, ribosomes, endoplasmic reticulum, microvesicles, cytoplasmic granules, a peripheral ring of microtubules, they can be seen along with the canalicular system in contact with the extracellular fluid. There are basically two types of granules, alpha granules and dense granules. Okay. So alpha granules, they have platelet derived growth factor, platelet factor 4, fibronectin. Dense granules, they have adenine nucleotides, serotonin, etc. You can also see the plasma membrane. Okay, it contains phospholipids and a coating of glycoprotein, and the membrane of platelets, they have receptors for ADP, von Willebrand factor, thrombopoietin, collagen, fibrinogen. Now about the synthesis of platelets. The process is called as thrombopoiesis. Now it starts from the progenitor cells. They differentiate into megakaryoblast. Megakaryoblast, they form promegakaryoblast. From that, there is formation of megakaryocyte and ultimately platelets are formed. Okay, so the entire process of thrombopoiesis, it takes place around 10 days. Now once these are formed, we will discuss about the variation in their count. When there is increase in the count of platelets, the term is thrombocytosis. It can be seen when the person goes to high altitude during exercise. These are the physiological conditions. Okay. In case of splenectomy also, we can observe increase in the count of platelets. Decrease in the count of platelets, it is called as thrombocytopenia. In this condition, the person tends to bleed and bleeding occurs from small capillaries. Decrease in count, it can be seen if there is bone marrow suppression, when there is exposure to certain kind of drugs that can cause bone marrow suppression, radiation exposure and splenomegaly. It can lead to thrombocytopenia. And there's a particular condition where thrombocytopenia occurs. Okay, what is that condition? In case of dengue. Okay. Now about the functions of platelets. The first and foremost function of platelets is hemostasis and blood clotting. Hem means blood, stasis means to stop. So these are involved in the arrest of bleeding by formation of a coagulation plug at the site of bleeding. They release clotting factor 4 and synthesize clotting factors 4 and 5 and 11. Once the clot is formed, it gets retracted. Okay, what is retraction? Once there is formation of blood clot, within few minutes of its formation, serum oozes out and clot becomes 1/3 of its initial size in around 30 to 60 minutes. Then the storage function, 5-hydroxytryptamine, it is stored in the platelets, which is released once these are disintegrated. Last one is phagocytosis of antigen-antibody complexes and some viruses. Thank you very much for listening. In the next video, we will talk about hemostasis and the clotting pathways.